initiating this wonderful series of the Science Dialogue series and introducing all of us with the new researchers, new scientists, new academicians with every episode of the Science Dialogue series. So, first of all, thank you to you. And now it's time to introduce our today's speaker, Dr. Shuvomoy Banerjee. Dr. Banerjee is an excellent researcher, academician who has excelled research in the science. He has done his postdoc from the University of Pennsylvania.
Click on your camera, Banerjee sir. Yes, so that people will know you. Yes.
Okay.
Yes, madam, please.
Okay. So it is my pleasure to introduce Dr. Shuvomoy Banerjee, sir. So he is presently working as an assistant professor in Department of Biotechnology and Bioengineering, Institute of Advanced Research. His expertise or his area of domain of the science, which basically belong to the cell biology, molecular biology, and related to the immunology. Then, to introduce him as a researcher, he has been a postdoc fellow at University of Pennsylvania School of Medicine, then at City of Hope Beckman Research Institute, from where he has done his postdoctoral studies. Then he has been a former assistant professor in Amity University, Noida campus, Delhi NCR region. He has vast experience in the field of research as well as in teaching. His area of expertise in research is basically related to the modern day topic, which is related to the apoptosis in cancer. So his expertise in cancer biology, immuno-oncology, tumor biology, related to that, biosensing, drug discovery, protein engineering, as well as molecular biology. So these are the expertise area of research for him. He has vast experience of teaching to the students, to many students, in the field of this immunology, virology, molecular basis of the disease, medical microbiology, cell biology, biotechnology, various aspects, and other life sciences course. If we talk about his expertise as a scientific community, he has been an excellent in the scientific content writing. He has many health communications in various leading platforms. He has been associate editors and the member of editorial boards in various reputed international and national journals. So this is just a brief introduction about the Dr. Shuvomoy Banerjee. I hope today's session of the cancers and the role of apoptosis in cancers might be a very interesting one to everyone, because personally I also like this topic very much, and I'm really looking forward to the session of today, that how we are going to get better insight about this topic of apoptosis in the cancer biology. So sir, now I am handing over this meeting to you, and let's start the session. Gopalji sir, anything you want to add?
Yes, just I want to add one thing for participants. Please don't ask feedback form after one day or after completion. So keep watching that chat box. I will give you this Google form feedback form after 30-40 minutes. So you must attend complete lecture, then only you can deserve e-certificate. If you have just registered and you are asking after one day, please give me certificate, we have attended. So keep watching chat box, please. I will give you randomly. Some people join after 40 minutes, 50 minutes, and then they say, sir, certificate milega ki nahi, certificate chahiye, I have attended this, and a screenshot. Then it's difficult for me. So attend complete lecture. I will give you Google form, feedback form in the chat. So after completion, during discussion, you have that link access. You can fill this form, and after few days, generally I send clubbing 2-3 webinar, and then send certificate. So please attend completely and then ask. Don't only just join and then leave, and then after one day you ask certificate. That is not good. I discourage this trend. Our purpose is to disseminate knowledge and improve overall knowledge pool of our society. Don't only to give certificate. And so just reminding you all. Thank you, sir. Please start, sir.
Okay. Thank you. First of all, I like to thank Dr. Gopal for kindly inviting me to deliver my lecture in this science session. And also I like to thank Dr. Poonam for very nice introduction for me. Basically, I like to say one thing. Today, especially, I made my slides for the science enthusiast and the students. So I deliberately just delete some slides which have very complicated scientific data, or means of my research works, and put relevant results on it. But basically I will try to make sure that the session will be engaging, and it will mostly talk about the how the apoptosis is going on inside a cell. So it's mainly for the students and science enthusiasts, and maybe I will come up with another session for the faculties. Okay, so let's start today's sessions. Just sharing my slides now. So the slide visible?
Yes, it's opening. Yes.
Yeah, okay, fine. So let's start my talk. My talk actually on the apoptosis in cancer pathogenesis. So first of all, we are seeing the beautiful petals are falling. The specific term apoptosis is a Greek word, and came from this specific phenomena with the fall of petals. It's a kind of metaphorical word that implies on the fall of petals, and ultimately the apoptosis, the term came from this concept. When the autumn comes, then the worn out old petals falls and the new petals are come from the bud. So actually replacing old thing with the new one is the concept of basic apoptosis. Then basically apoptosis is a kind of natural phenomena or natural kind of cellular phenomena, and it has diverse function. Number one very interesting function is apoptosis defines the shape of our body or the morphology. So morphological shaping or morphological manipulation first done by the cellular apoptosis. Here we are seeing the development of the mouse paw, and in between the fingers there are several set of cells, and these cells are deliberately worn off, they are dying, and this specific dying of cells in a specific region ultimately give rise to the specific shape of our body. Here it is the mouse paw. So elimination of some kind of biological material like the cells by the specific phenomena of apoptosis ultimately give rise the new shape of our body. So the shape forming phenomena is everywhere. Our nose, our toes, our fingers, our hands, everywhere. The apoptotic mechanism is very active. So apoptosis definitely a very crucial and biological function, and that involved in a specific growth, tissue homeostasis, and the maintenance of the healthy cells. So there are three major functions. Number one, I already discussed that the cellular arrangements and the cellular organizations they are influenced by the apoptotic process, and make sure that the specific cellular organs are doing their functions well. Also, apoptosis can regulate the cellular number. Every organ, every tissue maintain the specific cellular number, and such kind of maintaining the cellular number is basically depends on the phenomena of cellular homeostasis. So cell death and the cell proliferation should be balanced. We will talk later more in this phenomena, but apoptosis mainly playing some crucial role for the cell death, and it is keeping the very well balanced to to maintain the cellular homeostasis. Also, apoptosis performing very important function like elimination of the abnormal cells during the infections or any kind of disease pathogenesis. When the tissue is injured, when the tissue is actually affected by the specific pathogens, the cells are dying. Okay, and during this process, some cells may behave abnormally. Some cells get the mutations from the inherent stimuli, but the thing is, due to the activation of apoptosis phenomena, there will be a very fair elimination process of abnormal cells during any kind of pathogenic condition. Okay. Next, we can say very interesting features, that is apoptosis actually playing a very important role in the immune functions. Immune functions, immune modulation, and the apoptosis is a very important player to maintain the basic number of cells, as well as in context of the immune function, it maintains the tolerance of our body, and also other hand it maintains the immune response. Like if some foreign invasion happens, means if some foreign pathogenic invasion happens during the infection or the tissue damage or the inflammation, there are so many immunological processes are get activated, and basically this process sensed by specific set of genes, and ultimately it give signal the phagocytes. Okay, and this specific set of phagocytes ultimately activate the T lymphocytes to get rid of the actually infection and neutralize the specific infection. But this has two consequences. Number one, if the T lymphocytes are continuously activated, it may possible some malfunctioned lymphocytes may attack our own body cells, our own body tissue, and it will create problem, and it will create problem for the autoimmune disorder. Okay. On the other hand, if the situation is very genuine, like really a cell is invaded by the foreign pathogens, or it is getting owned out by the due to malfunctioning of some cellular organs, so it need to be destroyed. So immune response would be activated and cells will be destroyed by the T cell response. So there should be a fair balance between the tolerance and the immune response of our body. Here apoptosis is doing a very, very crucial role. So in a nutshell, we can say that abnormal lymphocytes, mutated lymphocytes, which are doing very adverse role for the autoimmune disorder, these are eliminated by the process of apoptosis or the programmed cell death. On the other hand, which cells are actually infected and ultimate cellular fate is not good for their survival, they have mutations, they have abnormal chromosomal quantity, that should be eliminated by the apoptosis also. So apoptosis will playing a dual role in cellular immune functions. In one hand, they are taking care of autoimmune actually response to save our body. On the other hand, it is destroying some kind of malfunctioned cells, some kind of infected cells to get rid of the foreign infection or to get rid of the disease pathogenesis. So immunity and apoptosis is doing a very integral part in this situation. Apoptosis, I said earlier that it plays a very crucial and important role in the tissue homeostasis. Very interestingly, being a natural uh, entity, means a biological entity, we are continuously exposed by the external stimuli, very dangerous external stimuli like UV, radiation, chemical exposures, and all that. These are all called the xenotoxic insult. And due to this exposure, our DNA getting affected. They are breaking. And this process is actually happening continuously. So the DNA breakage can be sensed by the cells. And these cells, either the DNA may be repaired with the DNA repairing machinery in a cells. Otherwise, if a specific set of protein things, I will talk about this more, because there are so many sensors in our cells. They actually sense, they actually determine the fate of a cell, that the cell will die or cell will survive. And when the sensor actually takes the decision, final decision, that cell, let the cell die, then the apoptotic machinery is getting activated. And with the two major pathways like extrinsic and the intrinsic pathways of apoptosis, generally this kind of abnormal cells or the infected cells are actually eliminated from our body. So tissue integrity and tissue homeostasis is very important, because every tissue should maintain their specific cell numbers to integrity, to make their integrity. And also, you know that from the genetical perspective, every cell should maintain, every species maintain the specific chromosomal number. If anything happens, like the duplication, ploidy, or anything, that ultimately influence the phenotypic expression of any kind of cells. So that should be a very problematic issues, and these sensor molecules ultimately take the decision that the cell will die or the cell will survive. And in context of the tissue homeostasis, apoptosis comes forward and eliminate the problematic cells. And infection, generally during the infection, any kind of pathogenic infection, it may be parasitic infection, it may be viral infection, it may be bacterial infection, diseased cells or the non-repairable cells, or the cells which are very harmful and mutated, and the cells which are accumulating some kind of toxins, the cells which are basically accumulating some kind of proteins that will create some abnormal phenomena or abnormal signaling, that should be eliminated by the apoptosis. So generally we see in different disease condition, in especially the pathogenic disease conditions, some infectious disease conditions, so where our body cells are infected by the virus and other pathogens, the cells are dying very fast. So in actually experimental condition also, if we just give some virus or just put some virus in the cultured cells, you will see the cytopathic effect, right? So such kind of effect is going on due to the onset of the cellular apoptosis. And this kind of phenomena basically follows the simple biochemical pathways, that the activation of the NADPH oxidase and the caspase 8. Generally, the pathogenic infections, during the pathogenic infections, aspase 8 the caspase 8 and the NADPH oxidase, it induce two things. Number one, it may induce the reactive oxygen species. We will show you more slides on it. But after the induction of the ROS, it may channelize its signaling efficiency in two ways. Either it directly activates the caspase 3. Otherwise, ROS induction activates the truncated bead, T bead, and it takes the pathway of intrinsic pathway of apoptosis, that is the mitochondria mediated pathway, and it ultimately activates caspase 9 and caspase 3 to let the cell die, and onset of apoptosis is going on. So cellular infection most of the time ends up with the apoptotic cell death. So I talked to you earlier that we need a very fair balance, because you know that the human body is made up of approximately 1 trillion of cells, and every day about 300 billion cells, approximate estimation of course, are produced as well as they die. Means near about 3 billion cells should die and new cells should take place every day. So there should be a fair balance. So this is the balance chart where we are seeing the cellular proliferation or the cell growth and cell death. Means cells will die, cells will grow. These two phenomena should maintain their specific balance. Sometimes it may not happen because cell proliferation takes place more than cell death, and this kind of abnormal situation we called cancer or the human malignancies. And sometimes apoptosis wins, means when we put some anticancerous drugs, when we put some anticancerous agent or any immunotherapeutic agent, then we will see that tumor is reducing. That means the cell proliferation is reducing, cell proliferation is going down, and the cell death is, we are getting more cell death, apoptotic cell death. So ultimately cancer defeats. So there is always a tug of war between the cell proliferation and the cell death, and cancer and apoptosis, because cancer always trying to reduce the rate of apoptosis, means the inhibition of this phenomena. That's why cancer always wins. But luckily we have some very powerful drugs now. We are trying to achieve several anti-cancerous drug with immunotherapy, gene therapy, and all that. So it may happen in very near future. We will get rid of from the cancer. So apoptosis, basically the game of balance. We talked about apoptosis, but this kind of phenomena first came to the public mind, and the public already knew the specific existence of this phenomena due to the great work of three scientists you know already. They are the Nobel Prize winner in medicine 2002, Sidney Brenner, Robert Horvitz, and John Sulston. These three great scientists first identified a very unique set of genes in C. elegans, Cinorhabditis elegans, in nematode. They work on this very tiny nematode to establish their hypothesis that how cells are dying, how cells are worn out, and is there any genetic correlation within this phenomena. So science just reported with a very interesting heading that the tiny worm lands a very big prize. Really a remarkable achievement by these three scientists who first defined the nature of apoptosis and the genetic correlation of apoptosis of human homolog genes in C. elegans. We will talk later. So basically, in this figure, you are seeing some color code, means some deep color, deep color and some light color shade. So basically, what is happening, as my heading suggested here, that apoptosis can be triggered by a range of internal and the external circumstances. Several conditions may trigger cell death or the apoptosis. Like most of the cell death is actually triggered by the hormone dependent change in tissues. In tissues, every time new cells are coming up and the old cells are going. So this kind of balance is going every time. This kind of balance is happening every time. So hormone dependent changes in tissue is the major crucial factor for inducing apoptosis. Next is the cell loss in proliferating or in population. When a specific tissue is getting some kind of genotoxic insult or kind of injury, the cells are dying, means abnormally. And the cell death here, here the cell death actually a stimulus for actually apoptosis. We will see this phenomena later. It is called the apoptosis inducing apoptosis, AIA. So such kind of cell loss in proliferating population ultimately triggers more cell loss. Another thing, exposure of the radiation or the response to the radiation or the any chemotherapeutic drugs can trigger the extreme cell loss. After chemotherapy or after any radiotherapy, the cancer patient actually experience a lot of cell death. They lose weight, their hairs already gone, skin are worn. So actually after radiation or chemotherapy, of course there will be a huge amount of the cell loss or the cell death. Also elimination of the abnormal cells, it's a kind of a signal that also induce the apoptotic signaling. And elimination of the lymphocytes, it's a very important thing. As I said you before, that the immune function is very closely associated with the apoptotic phenomena. So elimination of the lymphocytes, those who have the problem of the lymphocytopenia or any other low lymphocyte balancing problem, they face the such kind of cell loss or the cell death pathogenesis. And also the last is the normal development, means due to the color code you are easily understanding that such kind of trigger is a very minute trigger that we are actually exposing every day. That in normal codes some cells will die, some cells will grow. So it's a very normal thing, but still it is an inductive phenomena, means basal level apoptosis is going on every time in our body. So you may ask some questions. So sir, what is the morphological features or what are the biochemical signature of a specific cell death or the apoptosis? So specifically, when cells die with the induction of apoptotic signaling, they follow very specific morphological features. Like when cells are dying with programmed cell death, the cells will be scattered and they generally show the singleness. Okay. And another feature is the chromatin condensation. We will see the photo. The chromatin or the specific region of the chromatin are actually cut off with the endonuclease enzyme, and they will form a very specific pattern of band. So chromatin condensation along with the indentation of the nuclear membrane, we also called it the membrane blebbing. Nuclear membrane blebbing is another very interesting and the very strong features of cellular apoptosis and cytoplasmic vacuoles. Also, the formation of the cytoplasmic vacuoles. Sometimes cytoplasm may bulge out from the cells. It is called the cytoplasmic blebbing also. So we are getting two type of blebbing in case of BCD. One is the nuclear membrane blebbing, as well as we can see some cytoplasmic blebbing also. Nuclear fragmentations. A nuclear may be fragmented into several parts. Also, the cytoplasmic fragmentations and the apoptotic body formations. Cells are basically trying to shrink, and they appear as a fragmented bodies. And who will clear these fragmented bodies when a cells are dying, when a cells are already died, the debris are remain inside the interstitial space. So such kind of situation also induce a kind of signaling that attract the macrophage. So phagocytosis or the extrusion and the secondary necrosis may happen at this stage. And basically the apoptotic bodies are cleared off by the engulfment of the macrophage. So these are all the morphological features, the shape that is changing during the programmed cell death. But what are the biochemical features? So first biochemical feature, as I said, that when the cell scattered, when the cell show the singleness, of course there is a very specific point that the cell-cell junction proteins are actually detached from each other. So the separation of the cell from the neighbors means that the contact point already lost. And another thing is the upregulation or the higher expression of the endonuclease inside the cells. That ultimately gives the chromatin condensation, DNA already fragmented. Lamin disassembly means when a cell is trying to shrink, there will be a disorientation of the actin and the lamin cytoskeletal proteins, membrane proteins, and also the activation of caspase. This caspase activation is a central part of apoptotic regulation. Okay, and lastly, the expression of the surface protein molecules that triggers phagocytosis means that these apoptotic bodies actually expressing a specific set of proteins that behave as some kind of chemoattractant and that invites the phagocytes surrounding the apoptotic bodies to clear up the debris. It's a very interesting features that the brain cancer cell apoptosis is the scanning electron micrograph, and we took it from the and specific sources actually credited here. Very wonderful. And cellular apoptosis, if we see the cellular apoptosis, the morphological changes, membrane blebbing, formation of the apoptotic bodies, it's a sequential photograph from the cool CCD camera, that how the early apoptotic cell looks like, late apoptotic cell looks like, and the apoptotic bodies looks like. So it's the fate from a healthy cell to the apoptotic cells. So it's the sequential phenomena of cellular apoptosis and their change in morphology. So it's a very interesting. Sorry. Yes. So this beautiful hollow tomographic microscopic image, it's a level-free CCD camera captured image, which is actually showing the living cell apoptosis, and ultimately you are seeing how the membranes are shrinking, blebbing, and ultimately it forms the apoptotic bodies. So It may take 12 hours after the drugs, after the means administration, and ultimately you can capture this kind of beautiful image how a full-fledged cells is getting died. So it's a very wonderful image and showing directly the process of sequential steps of the PCD. Okay. Next, we know the cell death is typically occurs as a result of either necrotic cell death or a program cell death. Some people already asked me some questions, sir, what is the difference between these two things? The basic two things, even still today when we administer some kind of drugs or some kind of molecule to check the cell death, sometimes we confused what is going on. It's a truly apoptotic cells or it's a necrotic cells. So it is very, very crucial and very important to know, to distinguish between the apoptotic cells and the necrotic cells. So for students, I always say that necrosis involves the inflammation, is the tagline. So necrosis occurs when the cells die in response to injury, any kind of cellular injury, cellular stress, accidental injury, inflammation, and the specific features of necrosis will come up with the swelling of the cells, the bursting of the cells, rupturing of the cells, cytoplasmic content will come out from the cells due to mechanical injury. Some expression of the pleiotropic receptors also can be seen. I will talk to you later about these things. Very interesting phenomena to distinguish between this necrotic event. So basically, injured cells, it may be the physical injury or it may be chemical injury or anything that is related with the inflammation, ultimately give rise to the process of necrosis, and that is completely different from the apoptotic phenomena. And the apoptosis basically occurs when the cells are dying during the natural process of the tissue homeostasis maintenance and condensation, chromatin condensations and all that, without losing the membrane integrity. It's very important. We will see later. And membrane integrity completely lost in case of necrosis, but it is partially lost in case of apoptosis or the programmed cell death. And apoptosis is a tightly regulated phenomena. There is no question of induction of inflammation in this process. So it is the basic distinction between these two phenomena. So some people ask me, sir, there are so many types of cell death. We are confused, like autophagy, apoptosis, eroptosis, pyroptosis, necroptosis, means echinosis. There are so many things, mitosis. So each and every type of cell death has specific biochemical features, and basically we are trying to restrict our talk today with apoptosis. If Dr. Gopal permits in near future, we can talk more about this other kind of cell death. And basically cell death, mode of cell death also comes with the concept of apoptosis. So we can actually I am trying to convey you the classification of major three types of cell death that is going on simultaneously inside the cells. So number one, the major category is program cell death or the apoptosis. This apoptosis may induced by the chemotherapeutic drugs, immune system pathways, even ischemic death of the cells. Viral infection can trigger apoptosis. Already we saw in our previous slide. It's the type 1 cell death. type 2 cell death comes with the autophagic cell death. It may also induced by the chemotherapy, death infected macrophages, or also the immune function can induce the specific expression of the LC1, LC2 pathways that ultimately give rise to the autophagic cell death to get rid of the malfunctioned mitochondria, ER, and etc., the subcellular organelles. And the neuronal cell death sometimes trigger the autophagy. Retinal degeneration, very important physiological phenomena that ultimately trigger the autophagic cell death. It is a type 2 cell death. And the necrosis is the type 3. It is the category type 3 cell death. That also induced by the injury, inflammation, intoxication, liver injury, viral infection, or means also some bacterial infection also trigger the necrotic cell death too. So one very interesting things, if we claim that the apoptosis actually a conserved phenomena, genetically conserved phenomena, it's a evolutionary conserved phenomena, so we need to compare with the different model organisms. Means we are the highest organisms in context of the evolutionary tree. So we are mammals, so we have specific apoptotic related proteins. But if we see in case of the C. elegans, the first discovered in apoptosis in C. elegans, it's a invertebrate nematode. So C. elegans also have the apoptotic homolog proteins in their body. So they follow the specific gene regulation, gene expression, sequential gene expression for the induction of the cell death. Like CD4, CD9, CD3, or these all apoptotic homolog proteins are actually C. elegans related proteins. Their structure is more or less same, but there are some difference. But their specific homological domain aligns with the homological domains of the mammalian proteins like BAX, BCL2. You know already that the BAX, BCL2, cytochrome C, APAP1, caspase family genes, these are very important proteins which are always trying to trigger the apoptosis in vertebrate, in mammals. And if we see in invertebrate, lower invertebrate like Drosophila, I actually trying to say that 3 model organisms already scientists did a lot of work to establish the major apoptotic pathways in these 3 model organism, and they are trying to make the homology modeling, homology sequence modeling, but every time they are saying that some homolog proteins, at least some homolog proteins and their specific enzymes, either enzymes or the substrates are present in Drosophila, in mammals and the C. elegans to induce apoptosis. If time permits, we will talk more about the plant cell apoptosis and the animal cell. These are very interesting phenomena, and we are our team is trying.
We have conveyed participants about 1 hour. So in next lecture you can take other, but you have now 15-20 more minutes you can take.
Okay.
Yes.
Okay, sir. Thank you. So basically, we are trying to convey this message and this concept that in every phylogenetic tree, we are seeing the specific set of genes, specific expression of genes that are associated with the apoptotic functions. So apoptotic functions or apoptotic triggers through the intrinsic and the extrinsic signaling pathways. Students always say, sir, clearly mention these things, that what is the intrinsic pathway and what is the extrinsic pathways of apoptosis. Nature published this review image, And they clearly mentioned that the extrinsic pathway, 80% of the case, actually extrinsic pathway triggered by the external ligands. Okay, Fas ligand, FADD ligand, trail, and all that. These are collectively called the death receptors. Specific proteins, they are highly expressed in cellular membrane, and they are the specific receptors. Sometimes some tyrosine kinase receptors are also playing very crucial role for the induction of the specific apoptosis. But the thing is, how to remember and how to define this extrinsic and the intrinsic pathways of apoptosis in every disease or every means infection model. So it's a very interesting thing that whatever the induction is, the ligand may be 1,000, maybe several, but apoptotic phenomena always follow the specific pathways of extrinsic and the intrinsic induction of proteins that majorly follows the death ligand induced, death receptor induced activation of procaspase and the 8 and the procaspase 10 that ultimately cleaved with the caspase 8 and the caspase 10, and this caspase 8 and 10 induce the caspase 3, caspase 7 to finally induce the caspase 3. That is the executioner caspase. We called it the specific executioner caspase, and it meets the branching point. So extrinsic pathway induced with the specific onset of signaling which comes from the death receptors. And the intrinsic pathway, generally intrinsic pathway follows the induction of the intrinsic stimuli. Basically, damage of DNA, ear stress, hypoxic condition, deregulated metabolism, some kind of infection, especially viral infections. I worked a lot with the EBV virus, Epstein-Barr virus, and how this virus manipulates the intrinsic pathway we saw. The several viral antigens binds with the specific families of pro-apoptotic means proteins. They deregulate the Bax BCL2 ratio to inhibit the apoptosis. Either they inhibit, either they induced. It depends on their specific life cycle we see. And this intrinsic stimuli definitely covers the phenomena with the mitochondrial engagement, and such kind of Bax BCL2 ratio regulates the mitochondrial release of cytochrome C. And this cytochrome C is a very powerful molecule, powerful enzyme. This cytochrome C ultimately induces the formation of the apoptosome. Apoptosome is a combination of the cytochrome C, APOP1 and the caspase 9. This apoptosome finally induces the caspase 3, the specific executioner caspase, and cell death is going on. So extrinsic and the intrinsic pathway, but cell will die through the intrinsic pathway, and the whether it means it will die through the intrinsic or the extrinsic, it's not so straightforward. And several sensing molecules are playing a molecular switch. We call them the molecular switch, are playing very interesting role to get the decision that the cell will die through the apoptosis from the intrinsic pathway or the extrinsic pathway. So how we detect the cellular apoptosis? Very simple thing. It depends on the specific phenomena, that is the flip-flop movement of phosphatidylserine. If we follow the pathology, means any pathology book, we will see generally phosphatidylserine moiety lies in the internal membrane. internal cellular membrane, but during the apoptosis, it flips. So there are more phosphatidylserine moiety at the outer side of the cells, and annexin 5 proteins, we know it, very famous protein which has the very binding affinity with the phosphatidylserine. It binds with the phosphatidylserine, and if we tag any kind of fluorescent fluorochrome molecule like the FITC, rhodamine, and anything, so annexin 5 conjugated FITC will bind with the phosphatidylserine, and in case of apoptosis, it will show the fluorescence like apoptotic cells without disintegrating its membrane structure. It will show the simple flip-flop movement of the phosphatidylserine, and this kind of unique phenomena will be detected by either fluorescence imaging, either flow cytometry. Most of the people goes for the flow cytometry, and here we know after capturing this live cell with the annexin 5 FITC staining, we can easily see in lower right quadrant where you are seeing the early apoptotic cells, means they are annexin 5 positive, and PI or the 7AD negative, means their membrane integrity is maintaining, and no 7AD or PI dye is actually entering inside the cells. So there is no staining of DNA. So you will see the only signal from the annexin 5 FITC, and but these cells are specific cells which are showing the altered flip-flop phenomena of the phosphatidylserine, which is the basic marker of the cellular cell death, apoptosis. And if the time rolls on, you will see more cells in the right quadrant, upper right quadrant, which is actually the late apoptotic cells. Means here the PI or the annexin 5 is positive, as well as the annexin 5 is positive. So membrane integrity is lost. Some cells are still apoptotic, and most of the cells are dying. They are necrotic. They are showing the phenomena of necrosis. So two cell populations as a whole are showing the induction of the complete apoptotic process in your specific experiments. So you are getting the population of the early apoptotic as well as the late apoptotic cells. Also, apoptosis we can detect by the specific Western blot. Here you are seeing the two samples. We have the two samples. I take this picture from the app. I think AppChem. So here they are actually using the two samples, and they are treating the samples with the cytochrome C. One is cytochrome C null, and other is cytochrome C positive. So cytochrome C added samples are showing the induction of apoptosis. And what is the protein marker here? Pro-caspase-3 means the cleaved caspase-3. You are seeing very faint band with the pro-caspase-3 and the caspase-3, cleaved caspase-3. There are two subunits we are seeing. One is 21 kD near about, and another is a little bit higher. So you will see the cleaved caspase-3 here. And so in case of apoptotic induction, your Western blot clearly show the cleaved caspase-3 expression is extremely high. So it is your apoptotic marker by the specific Western blot. And also I told you that the DNA cleavage or the DNA fragmentation is a very unique phenomena for the apoptosis. So DNA is cleaved by the endonuclease activities, and if we run this DNA after treating our samples with any kind of apoptosis inducer, we will get DNA fragmentation pattern. Very powerful experiment it is, and it is clearly showing that DNA fragmentation pattern, actually it is the multiplication of 2. Okay, multiplication of 2 means two nucleosome apart it will cleave. Okay.
Hello, sir. Excuse me.
Yes.
Hello.
Sir.
So this Western blot, your marker is also, all markers are not. So this is purchased marker, molecular weight marker, protein
Yes.
marker, or it's your red.
No. I took the picture from the from the AppChem. So they are actually using their specific ladder.
Because you can see here 22.5 they have written, but that marking is not clear.
Not clear, right.
That is not good for purchased marker.
Yeah. That's
Because
why we rely on the rainbow marker that
Yes.
already we purchased. Yeah.
Means they are writing the molecular weight, but that marker is, that band is not
clear.
showing.
That's
Second
right.
Second thing, if it is SDS or this is tricine SDS, because 6.5 kilodalton in SDS, it's very difficult to see.
Yes.
They have
of course.
written, but that is not shown.
That's correct. That's correct. That's why it's the aspect sample figure that clipped aspect3 expression is high. But
I've seen so many times they are claiming about quality of antibody, but when we work in lab, that is not working in so many times.
That's right. Yes.
Yes. How many slides left, sir?
2-3 slides. I am actually concluding.
Yes, sir.
Okay. So already we talked about the apoptosis and apoptotic inhibition, pro-apoptotic agents, and we already talked about the cancer cells and DNA repair mechanisms. Also, the important thing is very here actually we need to see that the genomic instability triggers the two things. One is the induction of the oncogenes and the suppression of the tumor suppressor genes. Very common thing, but the balance between the oncogenic expression and the tumor suppressor expression also creates the problem. Means that if the oncogene expression is very high in cancer samples, we are seeing the huge cell proliferation and the inhibition of the apoptosis. So using the suitable drugs, we can induce the expression of the tumor suppressor to induction of the apoptotic process. Also, we told you about the several apoptotic proteins like the BCL2 family proteins, BAX BCL2 family proteins, PUMA, truncated bead, and these proteins are actually the major player of the intrinsic pathway of apoptosis. Also, intrinsic pathway of apoptosis mainly work on the regulation of the E53. Because E53 tumor suppressor expression is mainly regulated by the BAX BCL2 ratio. And if the E53 is null or the mutated in many human malignancies, then specific cancer will actually appear, and the apoptosis induction will goes down. Also, cancer cell inhibit the intrinsic pathway enzymes by modifying the cell metabolism. Means they have very strong influence on the glycolytic environment, glycolytic metabolism, and they induce very energy consumable process. Because cancer cell proliferation is very high, they need very high rate of metabolism. So they induce the glycometabolism is fastly, and they reduce the efficacy of the program cell death. Another thing I told you that induction of ROS is a kind of mechanism that induce the cell death. So reactive oxygen species ultimately that we can say that the superoxide ions, hydrogen peroxide, hydroxyl ions that actually came from the Fenton reaction with the superoxide isomers in presence of the SOD, hydrogen peroxide converted to hydroxyl ion and water. So these three things means the advent of the superoxide ion, hydrogen peroxide, and the hydroxyl ions. These three species are called the reactive oxygen species, and they continuously damaging our DNA. So DNA damage, also they damage the tissues also. So cancer actually trying to basically inhibit, but they have the dual theory. Sometimes it is said that cancer induce ROS for damaging the DNA, and they maintain this disease condition. And some theories are also appeared that no, actually apoptotic induction can be achieved by the induction of the ROS. So it is a kind of controversial thing, but generation of ROS is a very interesting phenomena in case of apoptosis induction. So ROS generation actually involves mitochondria, NADPH oxidase, peroxisome, the specific suborganelles also playing a very crucial role. Induction of the reactive oxygen species, and not only in cancer, it is implicated in the insulin resistance, mitochondrial malfunction, autoimmune disorders, and so on. And basically, our new drug, so many drugs are available in the market. So one class of drugs are continuously trying to reduce the ROS levels. They are called the ROS scavengers. And basically, they are inducing the expression of the NRF2, and it means NADPH, also the SOD, GPX. These are all antioxidants. They have a very influential ability to induce the antioxidants to reduce the ROS level. On the other hand, ROS generators are also there, like the oncogenes, even the high glucose level, high metabolism, hypoxic condition, they are all actually inducing the ROS to promote the cancer. And the cancer cell inhibit the intrinsic pathway basically to offsetting the oxidative stress mechanisms. So increased ROS means cell death, and decreased ROS means increase the survivability of tumor. And also cancer cells have evolved the very specific strategies to evade the extrinsic pathway. We already talked about. Some drugs are also we are trying to improve to target the intrinsic pathway, especially the BCL2 attenuation. So BCL2 inhibitors are currently people are actually using. Antisense oligonucleotides are using. Cyclin dependent kinase inhibitors are very strong, powerful drugs. And PI3 mimicking mimetic agents are also in market. And P53 MDM2 pathway modulator, which are the major player in the cellular apoptosis, we are also trying to improve the functionality. Also SMAC protein, if time permits, we will talk later. And trail-induced apoptosis means the trail inducer drugs are here. Also, we summarize these all things in my paper, which I tried to express about the apoptotic pathways by the gamma herpes virus. How this specific viral infection regulates the various nodes of apoptotic pathways. And cancer means already I told you that how you can detect the cancer means apoptotic cells with the drug-induced set of specific experiments. And single or the combination therapy means if you are using box, if you are using the chemicals, if you are using other drugs, so there are some limitations. So we cannot use the single drug at a time in this case. So we are generally go for the combination therapies. And the current total scientific community is focusing on the nanoparticle therapy, nano drugs, nano formulations, which are very efficacious. Okay, very efficacious to promote drug, to induce drug, to actually introduce very specific apoptosis inducing molecule inside the cells, to induce apoptosis, induce ROS, induce so many cellular machinery that ultimately kills the cancer cells. Okay, so future strategy for the cell death based cancer therapy. There are so many strategies, not only the specific apoptosis, induction of the ferroptosis, necroptosis, pyroptosis, even autophagy, apoptosis, and the other phenomena will be induced by either specific set of drugs. Thank you. And we am happy to take any kind of questions. Thank you.
Thank you very much. Now it's open for question. If anybody have any question, though we don't mute participants, they can ask question anytime because we commit that our lecture must be interactive. If they have any question during lecture, they can ask. Yes, Trilokji, please ask.
Yes. Hello.
Trilokji. Yes, Ramanna, you have raised hand. Unmute and speak. Okay.
Okay.
So Lakshmi, madam has asked, please suggest some reference book.
Okay. So there are so many reference book. I followed a specific reference book. That is the molecular biology by Baltimore. And another book is very important. You can use the genes series. I get benefited from this genes. Benjamin Lewis. Baltimore is enough. You can also check out the Cooper cell biology, very important. And there are so many books, but I prefer these three books. Yeah.
Trilok KNG, please join our YouTube. With the permission of a speaker, I will upload that lecture. We cannot give you PPT. So you join. I have given you all link of my YouTube and Facebook, LinkedIn, Twitter. So you can join and you can attend other lecture also. I have also shared feedback form in chat box. So many people have after given this, they have left after this filling the Google form because they know that after filling this feedback, they will receive certificate.
Okay.
So those who have not filled, please fill the feedback form I have given at 7:42 p.m. in this chat box. If any of you have any suggestion or any feedback,
I have not received, sir.
हेलो हाँ त्रिलोक नंदा जी क्या बोल रहे हैं
Sir, that YouTube link haven't got.
4 लिंक दिया है ना जस्ट चेक इन चैट बॉक्स प्लीज आई एम अगेन गिविंग यू एंड यस सब्सक्राइब यस सी द चैट बॉक्स
Yes, sir. The feedback link not received, sir.
please check at 7:42 if you were in the class you can access that 7:42 just scroll up that chat box please Trilok ji.
No, sir, it's not there. Just I was logged from 7 only, starting only. But I logged in laptop. Then
No, in laptop only it's visible chat box. Please see the chat box Google form at 7:42.
Initially I was in a mobile. Then I cut and due to technical issue I logged in laptop.
Okay. Just check. That's why generally I don't allow after 40 minutes. Now it's visible, Trilok ji.
No, sir. Can you
Please check in chat box. I have given chat box. Are you?
yes, sir. Yes, sir. Now I got.
So I have given. So you can fill, and if any of you have any open feedback, then you can also share. Any suggestion, any topic which you wish. There are so many professors are also, let me check. Okay. The professors have left. Fine. So Puna madam,
Yes, sir.
formally wind up this.
Okay, sir. Thank you again. It was really very interesting talk, and what I have myself learned about, or better to say, refreshed about my knowledge of this apoptosis and cancer biology, because this itself is a very vast domain for the research. Many researchers are devoting their life for this apoptosis only. So It was really interesting to me, as well as I hope that all the participants, they might have enjoyed the talk by you, and they might have been benefited from the content or from the talk that you have delivered to us in today's session. So thank you so much, sir. And we hope that in future also we may have some more elaborated session on the apoptosis or some other aspect of the cancer biology.
Yes.
So to get benefit from your experience, from your knowledge, and thank you so much. And again, a very yes, thank you again. A very warm thank you to Gopalji sir. As I said,
madam, for joining. Thank you, Shuvomoy Banerjee sir.
Thank you, sir.
Again, you can take 1 hour if you think that some important parts left. So,
Okay.
yes, whenever you have time.
Sir, actually, the is the work in apoptotic cell death is really vast, and I think I covered means only 0.1% of it.
Yes, it's difficult to cover complete research and other things going on in any topic.
That's right. Okay, sir. Thank you so much for your kind actually in the encouragement, and thank you inviting me in this lecture. I am really touched, and I always with students, faculties, and whatever they want, whatever they need from me, I will give. I will help every time. Okay, sir. Thank you, sir.
Thank you.
Thank you so much, sir.
Thank you, sir.
Still, any of you can unmute and speak if you wish. Otherwise, we are winding up. Chaitanya Mogal ji.
Sir, I would like to thank you for arranging this session to make us learn, and I also wanted to extend my thank to Banerjee sir and Poonam ma'am for helping us to understand the things in core, in depth. Thank you, sir.
Thank you. Thank you.
Thank you, Kashyap.
Thank you. Okay, so one people has asked about this PhD in industry.
Okay.
What are the scopes after PhD in industry? Okay, so we will discuss it separately. This is not one-liner answer.
Yes.
Anna?
Yes.
But
we need a separate session.
Yes, that requires separate session, and we have arranged one session whether we should go in academics or in an industry.
Yes.
So Jyoti madam had already taken one lecture on that topic.
Okay.
Again, we will arrange, because this is very huge topic, Srishti.
Yes.
There are so many criteria which will decide, Anna, whether you should go in industry or academics, and what are the scopes. So scopes are also depending upon your expertise. Yes. Suppose if you have only done counting of the leaf and you got certificate of PhD, then there is very less scope. So many people have just measured the thickness of the plant, stem and root, and then they got certificate of PhD.
Right.
So there are huge difference in the PhD holders' knowledge and expertise. No one can give you in one liner what are the scope, but there is huge scope in pharmaceutical company, in research company. There is huge scope if you have expertise on advanced topic of biology. Srishti, you have seen so many university gets blacklisted also because of selling the PhD degree.
Yes.
So only degree will not help. Your expertise, your knowledge, Hannah.
Yes.
That will decide scopes. Am I right, na?
Yes, of course.
Because I have seen the number of PhDs are very, very high, and they also apply for Pune post.
Yeah.
When UP advertised Pune position, what grade, so many thousands of PhD holders applied.
Yeah,
that means they are confused with writing PhD. Whether PhD में H capital होगा कि P capital होगा, I have seen.
Right,
so many BSc students, they are confused in writing full form of BSc.
Right. So actually education awareness is very, very important, and really sir, we need to make another session to discuss that what student will actually opt. after continuation of the PhD, and what kind of topic we need to choose in PhD. So sometimes it may help for the growing in the pharmaceutical industry based job. Otherwise, it will help in the future actually academics. So, what kind of topic you are choosing? It's extremely important. Very,
टॉपिक
very
बहुत
important.
इम्पोर्टेंट है एंड दे वन थिंग आई हैव ऑब्जर्व्ड इन people are joining PhD where they get certificate PhD award very easily.
Yes,
Meaning
of course.
PhD they don't devote time, but
No.
after PhD they start weeping and they are searching.
Yeah.
During PhD they opt the guide where there is minimum working environment.
Yes.
They don't
Of
want
course.
to work, है ना?
Quality control. Actually,
Yes.
quality control is very, very important.
Yes.
And, I saw a very interesting heading from actually USA. In my personal experience from the University of Pennsylvania, where I several time attended the PhD seminars, PhD counseling seminars, and they clearly mentioned that there will be no single boss will be your PhD guide. There will be a panel. So they have actually implemented. So the control of giving PhD and doing some work will not be handled by a single person. It could be handled by at least 3 panel members, and that could be fair.
In that case also there is RPS.
Yes.
So in RPS they have to present. But suppose in RPS you call your colleague and they will not oppose whatever you are teaching or whatever your students have done.
Yeah.
All the rackets are going on. That's why we have 140 crore population and we are purchasing something from France and England. Why? Because most of the people they are trying to get certificate. They are trying to purchase the certificate.
Purchase the certificate, right?
They don't earn. They just try to purchase.
They are actually compromising with the
Yes.
quality
During PhD,
education.
they try to wash the cloth of the guide. They try to purchase the vegetables of the guide,
That's true.
and then they get the certificate. That
Get the
all
certificate.
scenario we are discouraging this trend.
Yeah. Of course. They should be actually stopped, and some panel should be.
But that time, during PhD, they enjoy purchasing the vegetables of the guide and washing the
After
cloth.
that, what will happen?
Yes. And after that, when then guide is not available and guide is get retired, then they start weeping and then they are trying to search all these things.
Yes. Of
You
course,
must
of
devote time. You must earn the degree. Don't try to purchase the degree.
course. Yeah. Of course.
Hard working must pay. है ना?
Yes. Of course, sir.
एंड सृष्टि रिकॉर्डिंग नहीं है, उसका पीपीटी मिल जाएगा आपको. सृष्टि अग्रवाल स्कोप के लिए हम डिस्कस करेंगे 1-to-1, है ना, डिपेंडिंग अपॉन योर कैलिबर कैपेबिलिटीज. देयर आर सो मेनी स्कोप. सी one people beneath Jirla, he is trying to join now.
Yeah.
अभी भी लोग ज्वाइन कर रहे हैं. सी अब वो आएंगे, फिर वो जल्दी से हाथ रेज करेंगे, फिर बोलेंगे सर सर्टिफिकेट नहीं, हमारा स्क्रीनशॉट देखिए. So anyway, now I am not allowing him. Thank you everybody.
Thank you.
Thank you for participation also. So I am thankful to our research scholar student for joining, and they are trying to learn. That attitude is required if we wish to make our country Viksit and developed. Otherwise, only certificate holder people number is increasing. Gross enrollment will not help, and quality will help.
Of course.
So thank you. Good night. Good night,
Thank
madam.
you, sir.
Madam
Thank you,
Shuvomoy,
sir. Okay.
Good night, sir.
Thank you.
Good night, everyone.
Good night.